Okay, hello and welcome to my independent study tutorial session for those of you who can't make it. For those of you who don't know, my name's Ben Hatchell. You probably already all know that. Now I'm a human biology third year student. We're talking you through the effects of methylmercury on the human nervous system, how methylmercury is present in the environment, why I'm talking about methylmercury, not the other forms mainly, why I'm using hair as the sampling technique, and what the advantage is over other sampling methods such as blood, other sampling types such as blood, urine, why I'm using hair over those two, for example. So first of all, I'm going to be going through the background regarding methylmercury, why I'm investigating methylmercury in particular, then be going through an indicative schedule which will give you an idea of when you've got to be in for the particular sessions. I'm going to run them weekly throughout November. What the next one will be the first week of November. These will be weekly, subsequent weekly until we've had 5 sessions, and the final one will be at the first week of December. I'll then be going through some of the forms that you have to fill out, like consent form, risk assessment, selection availability, etc. I'll then be going through the significance of the study. I'll be talking to you a little bit about these nervous system conditions that you may get from methylmercury. I'll then go through the hair as a sampling type over the other types, and then that'll be pretty much it, I think, after confounded variables. So first of all, methylmercury is one of the 3 main types of mercury. There are other types such as mercury salts and phosphorylated mercury. I'm going to be mainly focusing on these 3 types. So organic or methylmercury, as we can see here, is the main form we get from seafood. Okay. So inorganic mercury can be reactive gaseous mercury, Hg2+, or HGO, which is the same as the elemental form listed at the base of the page. So the main form, Hg2+, is mainly found within water, and it's the main form of inorganic, which is found in water. The other form, HGO or elemental, is largely found within the air. The reason why Hg2+ has the name reactive gaseous mercury is because it reacts with the water to actually be accumulated within the water. And so I wouldn't be confused regarding its name reactive gaseous, that it's found primarily in the air, because it's primarily found there, but then it reacts with water, and so it's the main form which is found within the water that we're interested in, because that's where the seafood and the fish live. They're not actually called seafood when they're in the sea anyway. They're just called crustaceans and things like that. So so the elemental form, like I say, is present in the atmosphere. So why am I investigating methylmercury in particular? One of the main reasons is because it can actually penetrate the blood-brain barrier. Elemental and inorganic. Elemental is thought to be able to penetrate it slightly, and obviously this is particularly bad in children, as is the effects of mercury in general. In children, their nervous systems are still developing and still in infancy, and therefore they're more likely to be damaged by these intoxicants. So HGO, the elemental form, it can be emitted from both fossil fuel burning stations, and it can be emitted from volcanoes. Also, the Hg2+ or the reactive gaseous mercury can also be emitted by power stations, and phosphorylated mercury we don't really need to worry too much about because it's not one of the main 3 types. So as we can see from this diagram again, like I was saying before, HGO is largely found in the air. A lot of reactions can take place. Within the water, the main reaction is various forms, HGO and Hg2+, can be converted to methylmercury. This can also be gained from the soil, but it's largely converted in the water to methylmercury via things called methanogenic bacteria. It then bioaccumulates up the food chain through like plant life, like phytoplanktons, which then get eaten by small primary consumer fish, and it gradually is bioaccumulated up each subsequent trophic level, where the rate of detoxification through the glutathione pathway within the liver is lower than that of the rate of bioaccumulation, so therefore it becomes a more and more serious intoxicant as it moves up the food chain. And what this means is that larger fish have more of an impact to their nervous systems than smaller fish, and this is why we get more methylmercury from the meat of larger fish than smaller ones. But also, it also means because this methylmercury can actually impact the nervous system of the fish as well, it means that the ones which have more mercury are actually more likely to be caught, because obviously their nervous systems aren't working as effectively, and therefore we're more likely to actually have more of a problem because of that. So that actually confounds the problem essentially. It can also be obviously it can also be attained from the soil as well, this methylmercury. and soil is actually one of the main sources. It depends on the sort of properties of the soil, how many cations and anions it has, the charges within the soil. But anyway, I'm not going to go into that too much. So methylmercury is the organic form of the three, and it's got more of a nervous system impact, which is negative, than the other forms. Inorganic cannot penetrate the blood-brain barrier, and elemental has been shown to have less of an impact, and is thought to penetrate slightly. In younger children, all forms of mercury are more likely to penetrate and are more likely to have a more detrimental effect when they actually get into your brain and central nervous system. As I said before, it's found in fish and seafood. Large fish have more. Things like large predatory tuna will have more, in theory, than smaller mackerel, although there was a study in the Fiji Islands which said it's the other way around. And this may be due to these other factors, which I'll discuss some of the environmental ones. So you might have more factories around a particular area. You might have more volcanoes on a particular island. You might have more oil ships which are dumping oil into the ocean. You might have more pollutants for other reasons, and because of that reason, there may be more things that have a detrimental impact to the health of those fish. So there may be a lot of reasons why they have increased methylmercury from the environment, essentially, and not necessarily just due to that bioaccumulation, the effects of that bioaccumulation. So the big three fish they make up 71% of the fish meat consumption within the UK, are things like tuna, cod, and salmon. There are also prawn and haddock, which are also largely eaten in the UK. On my fish seafood consumption diary, I've labelled all of these in separate categories, and each category of fish corresponds to each column, whereas each row corresponds to the particular days in which you're consuming that fish. I'll talk you through that, obviously, when I actually give you that diary to fill out. I'll explain that to you. I can even give that in person. Obviously, I can put it on Facebook, and it's got some very basic instructions on it anyway. But just to mention, drained weight is what I'm interested in. If you're having tinned fish, if you have tinned fish, for example, a tin of tuna, on average tends to be around 400 grams total weight. I'm not interested in total weight because methylmercury bioaccumulates within the fleshy tissues primarily and not the lipid oils. Therefore, it's more likely to be present within that fleshy tissue. So I'm interested in the drained weight, which is around 240 grams for the average tin of tuna. Indicative schedule I'm going to be going through with you now, which is why it's highlighted, and this is when you have to come in. So like I said, for weekly sessions throughout November and the first week of December. You also come in for the final session, which is where you'll provide your small hair sample. I say you'll provide it then, you actually collect it previously to that by storing hair up in your hairbrush. You bring in that hairbrush, and that hairbrush is where I get the hair sample from. Okay, I'll process that, so obviously you don't have to worry about the processing of that, but essentially all you need to do is put your hair sample, which you've collected, within this sampling bag. I'm more than happy to do that for you, obviously. Then I have to leave the room because you'll write your participant number on your bag, so that's what, to maintain anonymity. And then you will place that within a box when I'm out of the room, and as long as they're all sealed, all the hair samples are in each bag, then that's absolutely fine. Obviously check that they're all sealed and that they're all the numbers from 1 to 30 are in there. If that's the case, your participant number obviously can be ticked off, and I'll know that you've done it, so that's absolutely fine. So that's essentially how it's going to work in the first session of December, which is the final session also. So part 3, I'll normally be discussing Q&A, but obviously because obviously you was unable to attend due to other commitments such as work, for example, we're not going to worry about that. Obviously you can ask me face to face some questions if you want the answer. Obviously you can ask me via email, happy1_13@uni.walk.ac.uk, or you could also alternatively message me on Facebook privately. So anyway, so indicative schedule, we have our tutorial session, not today, it's on this Wednesday that's coming up. That will be one of 6 sessions. You have 5 sessions, 1 weekly throughout November, the final one early December, which makes up 5 out of the 6 of the total sessions, obviously excluding tutorial, which makes it up to 6. The session shouldn't in theory take very long, should take, I've got written down there 40 minutes, in theory it should probably take even less than that, because each task, like when I did the task, to give you a rough idea, 1 to 2 minutes probably per task, and then you got 7 tasks, so the actual tasks themselves shouldn't take under 20 minutes, but therefore obviously actually getting in there, like getting settled in and all that might take a little bit longer, and actually collecting in the data at the end and everything like that might take it up to the 40 minutes. So that's essentially why I've written that there. Obviously, the lab's going to be booked for a far longer time, so at the end of each session, if you have any questions, you can address those directly to me. So anyway, we're going to go through the forms now that you actually have to fill out. So do not panic. I understand you're not going to be given these at the tutorial session because you're unable to make the tutorial session. But you have to select a number from box 1. You have to write your name on the reverse of that and place it in box 2. And these boxes might not be boxes, they might be bags, but these will be present in the Sheila Scott Building 004. If you don't know where that is, ask main reception. They'll know where the block is and point you in the right direction. And once you get there, then you can write your name on the reverse of that, place that within the box while Christian's there. So hopefully within working hours that should be more likely that he'll be there, and hopefully he'll be able to do that for me. I'm pretty sure he will. Obviously, if you have any problems with that, like, come and see me. I'll try and get those boxes and bags there as soon as possible, and I'll try and notify you in person once I've done that. Obviously, I'll put a post on Facebook anyway so that you all get to know, because obviously I'm fully aware I don't see all 30 of you on a daily basis. So anyway, so selection availability criteria form. This is the same as the exclusion, but it enables me to gain information regarding when you're actually likely to be available. This might not be that applicable now because I'm fairly confident I want to do these sessions on a Wednesday at lunchtime, but it may be that I can arrange a session for those of you who work quite a lot on a different day, such as a Monday, for example, around lunchtime. So anyway, consent forms. There's 2 copies of this, and there's one which is for me, and there's one which is also. There's 2 which are for you, but you hand one back to me and you keep one. Is essentially what I mean by that. And that's just a simple consent form, so it should have like signature. Might have a few other bits of information to fill in, but they're only basic information like signing, name, perhaps dating. So anyway, so you have the risk assessment, which you only fill out 1 row of, and there's 30 rows corresponding to each of the 30 of you, and you'll be provided with that at your first nervous system test session, because I only need it before you actually start doing the practical stuff, which it's called practical. All you're doing is some paper tasks, so that'll be quite simple to do. But that will be at the subsequent nervous system weekly sessions, and I'm not going to put that on Facebook because purely for the fact that it would be complicated for you to coordinate for yourselves, like what copy of it has all the numbers on. You'd end up just posting on my group constantly, which I don't know if you're even able to do, but if you are, then you'd end up 30 posts on there with 1 extra row completed every time, which is an impractical way of doing things. So I'd rather you just complete that on the first weekly test session of the nervous system, which is in the first week of November at some stage, subject to ethical approval, which I will let you all know via Facebook when I've got that. So anyway, seafood consumption diary, I can provide you with that over Facebook anyway. That's absolutely fine. Only 15 of you need to do that because half of you are seafood eaters, half of you are not. I understand some of you may eat it occasionally, but how I've defined it when I was selecting you is how you're defined, and you should in theory all know which group you are in based on like whether I've spoken to you directly. Like, do not panic. Supposing you don't panic. You can obviously message me, or you can fill it out on the form. But ideally need to know that you haven't changed that, because I've listed everyone down with regard to what category they're in, which I'm obviously going to destroy once you've all got the seafood consumption diaries handed in. But I need to know. I need to know that you haven't changed category, otherwise the sample numbers might not be even, and you might end up with sort of a 16 and a 14 rather than 15. I want to ideally keep it as what it is. Supposing your status changes, for example, supposing you have a craving for prawn sandwiches halfway through November and you're in the non-seafood eater group, it doesn't matter by that point because I've already assigned you your group. You still won't have to do the diary. That's absolutely fine. So anyway, so you take 1 copy of the participant information sheet. I understand from some other people's studies that you don't actually all probably want a participant information sheet. I've spent a lot of time on it. I'd appreciate it if you do want one, because it'll give me satisfaction in providing that for you. I'll obviously put that on the Facebook group again, so you can have that if you want to, and it is quite a good read, to be fair. So anyway, 5-minute break. We're not going to have a 5-minute break because I'm going to roll the whole way through this because I want to get this done now, but obviously we can see this guy presumably is having a far longer break than 5 minutes. So anyway, part 2, significance of my studies. I've spoken with regards to methylmercury in fish, mercury in the environment. I don't want to bore you to tears too much, but there is negative impacts, as we've seen, of methylmercury on the human nervous system. I'll go through some of those with you now. Hair versus saliva samples. Some people use blood, some studies use blood, some have used hair. These are the most two effective techniques. I'll discuss why hair's better. So anyway, so chronic fatigue syndrome is one of the nervous system issues which is caused by the neurotoxicity of methylmercury crossing that blood-brain barrier. It affects around 250,000 people within the UK are affected by it each year, and this is also known as myalgic encephalomyelitis, which is quite a mouthful, but there are disputes over the name, so you can just call it chronic fatigue syndrome because it changes its name quite regularly. It may well change back to chronic fatigue syndrome. The reason why they changed it from that is essentially because methylmercury doesn't just inhibit you in terms of your sleep cycles and circadian rhythms, it actually impacts you in far more different areas, things like mental concentration, it affects your memory, it affects your visual processing, it affects, like I'm testing, like number letter sequencing. It can, in children, it can lead to an increased risk of things like cerebral palsy. It can cause huge language difficulties, difficulties in speech, it can cause muscle weakness. There's a huge range of different things, and this muscle weakness again is related to cerebral palsy, which is an issue with the muscles itself. Okay, so anyway, so myalgic encephalomyelitis is what it's sometimes known as, and anyway, some of the symptoms are malaise, lack of sleep, and cognitive issues, which I've just mentioned a few of those with you. So what do I call it? I don't know. I wouldn't worry too much. Just call it chronic fatigue syndrome if you want to. That's absolutely fine. But like I say, it's one of the main nervous system issues which is caused by methylmercury. There is a video. I won't worry too much about watching the video, but it's quite nice in the sense that it shows you what's actually happening to your nervous system once you consume that methylmercury. Once it gets into your nervous system, it's actually impacting the growth of the sheaths that surround your axons, which are what takes the impulse away from your cell bodies and actually projects them. So that's quite a nice video. And it essentially shows how this tubulin is affected, which is involved in a sort of neuron synthesis. Okay, main regions within the cell that are affected are things like cell membranes, plasma membranes on the outside that surround your cells, and the mitochondria, which is the energy centers of your cells. It affects astrocytes and neurons, because neurons aren't the only central nervous system cells. We also have astrocytes, which are involved in regulation of blood pressure. So this can actually lead to things like cardiovascular issues as well. So anyway, nervous system test impact. How am I testing it? You're going to do tasks regarding things like reaction time or memory, which are thought to be the best indicators. I'll carry out 5 repeats over 5 sessions, which are 1 each week, and then there'll be a range of tests used each time. There'll be 7 each time, and these include number sequencing, letter sequencing, number letter switching, motor speed, reaction time, and visual memory. I hope you'll enjoy doing these. These really aren't laborious long tests. They're very quick, and you'll hopefully have some fun doing it and seeing how good you are at them. They're not just plain boring reaction time tests. That there is quite a lot more to it than that. There's 2 which are interactive. So for the first weekly session of November, I do require you to bring in your laptops. Remember to bring in your laptops then, because supposing you don't, you'll have to all be crowded around my laptop at the front, which I'm providing the presentation from. And although it will be only a very quick presentation for my nervous system test sessions, obviously it means that it's going to take a lot longer if I have a queue of 30 people not able to do it on their own laptops. Okay, you can obviously do that in your own time. That's not so much a problem. Well, actually, I'd rather you didn't do it in your own time so I can control the conditions, because obviously I don't want anyone like fixing results or anything. So I ideally do want you to do it when you're there. So you do need to do it once you actually come in. Okay, so obviously those of you who can't attend, that isn't a major problem because I will not include those results for that week. So then moving on to methylmercury within hair, the elements including methylmercury are incorporated within the growth phase anagen within the hair. 90% of the hair is within this phase, anywhere around 90,000 hairs. There's around 100,000 hairs on your head. And 100 of those will be in the final phase of life telogen, which they can weekly be pulled out. They don't even have to be pulled out. Essentially, this is why I'm collecting samples from the hairbrush. When you brush your hair forward for 60 seconds, it's likely that you will lose 10 hairs on average from a 20 to 60-year-old. Most of you are within that range, some of you are 19, and therefore, obviously, it may be slightly different, but it should be, as a whole, quite accurate. Regarding that, obviously that's a mean average. You might lose loads of hair in comparison to the person next to you, but as a sort of main average, you'll lose around 10 hairs per day. I need around 44 to 300. I'm going to take roughly 2 weeks, assuming it's 10 average per day. I'll ask you to keep the hair on your hairbrush for 2 weeks before you come to the final session early December. Bring along your hairbrush with you. I'll obviously take it off, put it in your sampling bag. I'll leave the room. You write your participant number on that sampling bag, you then put that into the box, and that's absolutely fine. That's all you need to do on the final session. Before that, obviously, it's the only the nervous system task. So until December, you don't have to worry about hair at all. And some papers use less hair than what I'm going to be requiring, and this is simply because they have a more sensitive sort of technique in order to process that. Hair is made up of 65 to 95% protein, which is significant because the keratin is the main type of protein which helps lock in that methylmercury within the hair, which means that it can actually last for a particularly long period of time. In fact, it's actually stable at room temperature for years, which means that I, in theory, would have years to process your hair samples if it wasn't for the Human Tissues Act, which means I need to process them within 1 week. But anyway, so you'll lose about 10 hairs within your hairbrush session, around 60 seconds forward. This is at home. You're just doing this when you're in the bathroom before you get ready to go to uni. Just remember not to pull off your hairs off the hairbrush every time, throw them in the bin, because Ben needs that from the second half of November, building up to December. And supposing you have too many hairs, great, doesn't matter, I'll sort that out. Supposing you have too few hairs, maybe you shouldn't have cleaned your hairbrush regularly, or maybe you just never lose any hair because maybe you're pregnant, which I obviously don't know, and therefore obviously you don't have to provide a hair sample if you're pregnant, because I believe pregnant women don't lose hair. So anyway, so 5mm hair samples, obviously this varies depending on how fine the hair is, how many you need. For really fine hair it will be around 333, for thicker hair types it will be around 41. But essentially, because you're losing a similar amount of hair, like 2 weeks should in theory provide a mid-range between the amount I need, so that's roughly on average what I require. So that's why I'm asking roughly 2 weeks before the final session early December. So from about mid-November is when you need to start storing that hair, keeping it on your hairbrush. Okay. So anyway, like I said, it's stable for years. Each month is represented by 1 centimetre of hair growth. The first 4 centimetres are the most reliable indicators of methylmercury exposure. 1 centimetre hair growth will have accumulated the methylmercury from the fish and seafood consumption of that previous month. So at the end of the month, if you cut off 1 centimetre of your hair, that hair would be representative of your previous month's methylmercury exposure. It's important I know that, so because I've sort of gained the information about whether you're a fish or seafood consumer over the last 4 weeks. Obviously, if you have been a seafood or fish consumer over the last 4 weeks, then that will show up in your hair. So it means that my study is valid and it will work, essentially. So hair sampling is good because it's non-invasive, it's got good response rates, and it's good for, and it's better than blood because it requires lower cleaning temperatures within the graphite furnace, atomic absorption spectrophotometer, which I'm using to process your samples after your final session, which you don't need to worry about at all. Basically, there are a few confounding variables, things like race or ethnicity, which are confounding it in the sense that they alter the bioaccumulation of the methylmercury within your hair. And there's omega-3 fatty acids as well, which actually actively benefit the human nervous system. Okay, and that's it. So thank you for listening to the presentation. Hope it's been useful. Like I say, I'll post all the relevant forms on Facebook. Hope you've learned something new. If you have any issues, contact me via email, happy1_13@uni.walk.ac.uk. And supposing you don't want to do that, you can contact me on Facebook. You can ask questions after the sessions, which hopefully I'll have ethical approval by then, which I will have ethical approval by then within the first week of November. That there'll be 1 session then. Because of some of your work commitments, you may not be able to actually come to every session, and I fully understand that. That's absolutely fine. What I'll do is I won't take the results from you that week. Supposing a lot of you can't attend, I'll try and arrange multiple sessions per week within November, and I'll try and sort that out sort of nearer towards the end of this week. I'll try and gauge from the selection availability form, which I want you to fill out ASAP. Actually, when you're free on that, there'll be some sort of section for you to fill out what days you're free for the sessions. Obviously that's the exclusion criteria form, so you just fill out a few bits of exclusion criteria, so the age you are, whether you're a seafood, non-seafood consumer, the length of your hair, that doesn't really matter anymore. I want you all to fill out that still, but that doesn't matter because some studies have used as short hair as 1 centimetre, so although the study I originally looked at required 3 centimetres, as long as you have hair and as long as it comes out in the hairbrush, I should be able to get sufficient sample size from your hair. Okay, so I hope I haven't missed anything out. I'll contact you with any further information that I'm required to give you via Facebook. If you have any questions, contact me. Thanks for listening. Have a good shift at work or whatever you're doing on Wednesday, and I'll hopefully speak to you soon. Goodbye.